Okay children, the next topic what you are going to study is virus-free plants. Now you have seen that most of the plants when they are grown in the soil, they are infected by different disease-causing pathogens like bacteria, fungi, virus, etc. So here this fungi bacteria, they can be, the diseases caused by fungi and bacteria, they can be controlled by using any chemical agents like insecticides, pesticides, herbicides, etc. But for the protection of viruses, difficult. So here we are using another method of raising this virus-free plants by shoot meristem culture, shoot tip meristem culture, because this meristematic tissue, they are free from virus infection. So here let's see the lesson. The field grown plants like perennial crops. So usually the perennial crops are infected by this disease causing pathogens. Perennial crops are what? The plants which have a lifespan of more years. Okay. They are infected by variety of pathogens like fungi, bacteria, mycoplasma, viruses which cause considerable economic loss. So when the plants they are affected by the pathogens, naturally the yield of the plant is affected, it reduces. So chemical methods can be used to control the fungal and the bacterial pathogens but not viruses generally. So here by the chemical methods we can control the other diseases, but virus to protect the plant from the virus infection is little difficult. So shoot meristem tip culture is the method to produce virus-free plants. So the best method of getting of virus-free plants is the shoot meristem tip culture, because the shoot meristem tip is always free from virus. So as we know, in the tip of the, that is the shoot tip and the root tip, we have the meristematic tissue. So here this meristematic tissue from the tip region, they are taken for this shoot tip culture. Okay, because the meristematic tissue, they are always free from virus. Now how it is done, that we are going to study here. Now before that, let's see here in the apical meristem, the diagram we can say 3. This is the shoot tip. In the shoot tip we have the apical meristem cells, and this shoot tip, they are protected by the leaf primordia. That is the they are the leaf initials. Yeah, they will later on develop into the leaves. As we know, the buds, they are protected by the leaves. So they are arranged in the form of a horn-like structures. Here you can see in this diagram. So here, so there are numerous leaf primordia is there. But out of that, 1 or 2 only is being used for the culture, or that also can be removed from this bud. So how it is done, the steps involved in this, we are going to study here now. So protocol for virus-free meristem tip culture. So first one, apical meristem tip with 1 or 2 leaf primordia are excised in sterile condition from the explant. So from the explant, the desired plant, what are you doing? We are taking the apical meristem, which will have 1 or 2 leaf primordia will be present. So this apical meristem tip, they are being sterilized. They are sterile conditions. They are sterilized by using the chemical sterilizants, by surface sterilization. Next, in vitro culture in 10 ml of solid MS medium supplemented with growth hormone. This apical meristem tip, which is in the sterile condition, they are incubated. They are transferred to the culture medium. So the culture medium, as we have studied, the most suitable culture medium is the MS medium. So here we are taking 10 ml of the solid MS medium we are taking, which contains the growth hormones for the growth of this apical meristem. Next, what we are doing, we are transferring this, we are incubating this. So first one, we have introduced the meristem inside the culture medium, that is called as the inoculation. Next, what we are doing, we are incubating it, so for the formation of the callus. So here this apical meristem in the MS medium, this this inoculation, they are being transferred to the incubator, where the temperature is maintained at 24 plus or minus 1 degree Celsius and is kept for 3 days in the dark. After that, it is kept exposed to the illumination, that is the normal fluorescence light is provided, which is the light intensity of 2400 lux. So as a result, what will happen when it is kept in the incubators, it will lead to the formation of the callus. That is, a mass of cells will be developed, and from this callus it will undergo morphogenesis. That is the here organogenesis is taking place. Organogenesis means the organs will be developed. What are the organs? The shoot and the roots will be developed here. So from the callus, some of the cells will develop into the shoot. Okay, and after the shoot formation, because of the hormones present in the medium, it will lead to the development of the roots. So here the young plantlets are ready now. Okay, now this, after the development of this plantlets, they are transferred to the natural condition, because they are now in the in vitro, in the laboratory conditions they are being cultivated, they are grown. Next, slowly they are exposed, step by step they are exposed to the natural condition, but in a shady region. When they had established them in the shady region, they are slowly transferred to the field where they can cultivate it. So this is the protocol, or this is the method for raising the virus-free meristem tip culture from the virus-free plants from the meristem tip culture. Next method is, the next what we are going to study is the conservation of plant genetic resources. So next is, so here as we have developed the new variety of plants, they are the plants, the natural plants also, their genetic material has to be conserved for the biodiversity and for the continuous supply of the food from these plants. So one method is the germplasm conservation. Germplasm means the living content of the cell. Living content. So germplasm conservation refers to the conservation of living genetic resources. It may be the pollen or the seeds or tissue of the plant material, tissues that may be from the leaf, stem or the roots. They are maintained for the purpose of selective plant breeding. So here the method, the need for this conservation is, we can use it for our research work, for maintaining the biodiversity, for our hybridization experiments, etc. So here this is done for the preservation in live condition and used for many research works. Germplasm conservation resources is a part of collection pollen that are stored in seed or pollen banks so as to maintain their viability or fertility for any later use, such as hybridization and crop improvement. So here this germplasm, they are being conserved in the seed bank or in the pollen banks, so that we can use it later for our experimental purpose or for the improvement of the crops also. Now, germplasm conservation can also involve in a gene bank or DNA bank. So here the genes of the plant can also be conserved. The DNA of the plants can be conserved for getting the elite varieties of, they can be conserved to maintain the biological diversity and also for the food security. Another method of conservation is the cryoconservation. Okay, cryopreservation or cryoconservation. Cryoconservation is also known as cryoconservation. Now, what is this method? Now, here the cell or the tissue, they are preserved at a very low temperature of minus 196 degrees Celsius. Okay, so when they are preserved, the cells, they are preserved at a very low temperature, the activity of the cell will be dormant. Okay, they will be reduced, so it can be preserved for a longer time. Now later when we want to use it, we can bring them back to the normal condition and we can use for a research purpose or for the needed purpose. So here cryopreservation is also known as cryo process by which protoplasm already we have studied, it's a cell without a cell wall, bounded by only cell membrane. So here the protoplasm can be preserved. Cells, tissues, organelles, organs, extracellular matrix, enzymes or any biological material, they are subjected to preservation by cooling to very low temperature of minus 196 degrees Celsius. So here this entire cell or the protoplasm or any biological material, they can be preserved at a low temperature of minus 196 degrees Celsius using liquid nitrogen. Now, at this extreme low temperature, what will happen? The enzyme or the chemical activity of the biological material will be totally stopped. So, when it is preserved, the biological material of the cell is preserved in this condition, extreme low condition, the activity will be completely stopped. So this leads to the preservation of material in a dormant state. So here the cell will be preserved in a dormant state. That is, the activity of the cell is ceased there. Now the cell is in a dormant state, but it is in a alive condition. Later, these materials can be activated by bringing to the room temperature slowly for any experimental work. So here, later these materials, okay, these preserved materials, they can be used. When we want to use this preserved materials, first we have to bring them back to the normal temperature, normal room temperature, and then we can use it. So it will be active that time. Next, another precaution that has to be taken before cryopreservation is these materials, these biological materials which are going to be preserved, they have to be protected by they have to be protected by using some cryoprotectants. Cryoprotectants means the materials which are used for preserving these these biological materials. So the agents that are used are dimethyl sulfoxide, glycerol or sucrose. They are added before the cryopreservation process. So these agents, they will protect the cell or tissues from the stress of the freezing. So here, to protect the freezing temperature, to protect the cells from the stress of the freezing temperature, these cells or the tissues or the biological material, they are treated with this dimethyl sulfoxide, glycerol or sucrose. So here this will protect the cell from any damage. Okay, so this is the cryopreservation, a very important topic. What is cryopreservation? Is a very important topic, children. And here you have studied and this method here. Hope you have understood all the topics taught today. We will continue with the next topic in the next class. Thank you, children.